Hello everyone, welcome to another video by IGCSE Passion. For the day, I'll be explaining Chapter 5 from 0610 Biology IGCSE, which is enzymes. Here are the points that will be covered in this video, which are like the latest syllabus for this subject. Let's start with our very first point. What is a catalyst? Here we have 3 points that should be mentioned when defining a catalyst. First of all, it's a substance, a chemical substance, that increases a rate of reaction without getting used up. Let me just highlight the important points. Here we go. So it's a chemical substance, that is number 1. Number 2, it increases a rate of reaction and it wouldn't get used up when it gets into a reaction. Next, what is an enzyme? An enzyme, here we have also some points that should be mentioned when defining it. So number 1 is that it's a protein. So it's made up of amino acids. Secondly, it's a biological catalyst. Like if you can't recall what's an amino acid or a protein, I highly recommend going back to chapter 2. I've explained it earlier in one of my videos. You can check it out. Wait, let me just point it out and say that this one is related, I guess, to chapter 2 if not wrong. Okay, it's a biological catalyst. Biological because it's made up of proteins. It reduces the activation energy. Some students may not know the difference between increasing and reducing activation energy. I'm gonna explain it in the upcoming slides, so stay in this video. Next, and it increases the rate of reaction without getting used up, just like normal catalyst. Increase the rate of reaction and does not get used up. Where do we use an enzyme? We use it in metabolic reactions, okay? Next, why is an enzyme important? Why do we use enzymes in our body? Because they control the reactions in a cell. So we have several chemical reactions happening in a cell, known as metabolic reaction. So they make sure that this reaction occurs quickly enough for cells to function so we can live. If we don't have enzymes, then humans won't be able to live, nor other living animals. Here are the actions that happen. Now, firstly, we know that the enzyme have like a certain shape, okay? Now it's made up of amino acids that are bonded together like in a chain, and then we're gonna have a complex structure known as a protein, okay? Now this is, let me use my laser pointer. Okay, now this one is what we call an enzyme. Now an enzyme would connect to a substrate. The red thing is a substrate, and how do they connect together? We have something known as a lock and key relationship. So who is the lock and who is the key in this relationship? The lock that we have is the enzyme, and you're gonna know why. And the key is the substrate. It's the substance that gets connected to the enzyme. Now the enzyme have an active site. Now this part that contains, like it is where the substrate can connect, is known as the active site. Okay? And it is complementary to a specific substance. Let me just point this out. You need to mention that it is very specific to a certain substance, creating an enzyme substrate complex. Okay, now when they ask about the actions of the enzyme, you should mention that it contains an active site where the substrate can connect, and then you're going to mention that it is very specific. Thirdly, you're going to say that they will make an enzyme substrate complex. Now, after you bind the substrate to the enzyme, a product will form, so it may break it or it may just connect it together. So as you can see in this case, it broke it into two pieces, and those two pieces are known as the product. Okay? We have two actions that can be performed by an enzyme. We have anabolism and catabolism. Anabolism is connecting particles, catabolism is breaking. And you should know what happens to energy. So energy is used in anabolism, catabolism releases energy. Enzymes and temperature. A rise in temperature increases the rate of most chemical reactions, but a fall in temperature slows down the rate of reaction. So here we have a graph that represents the reaction rate, and we have the x-axis as the temperature. You need to know the labeling for the axis, and then you're going to say that usually a rise of 10 Celsius will double the rate of an enzyme controlled reaction, okay? But the optimum temperature of about 37, it is the body temperature. So the optimum temperature for the body is 37. After that, what will happen? We're going to say it after it. So this is because of enzymes and substrate molecule are constantly moving using kinetic energy. Wait, let me use another color so you can focus here. Color coordinating can help you with focusing. Okay, so the reaction only happens when the enzyme and substrate molecule collide with each other. So an increase in temperature gives a higher chance of collision. So as the temperature is increased, molecule gains kinetic energy, so they move faster, and there's a greater chance of collision. So that's why the rate of reaction will increase. Now talking about the optimum temperature, about 50 Celsius, the molecule gain even more kinetic energy, but their action will slow down. Why is that? It would slow down because protein molecules starts to lose their shape. Like the bond between the amino acids will start to break, and so the complex structure of the protein will start being lost, and high temperature will cause a change in the active site. As a result, although there are more collisions due to the molecule having greater kinetic energy, the number of effective collisions will eventually get reduced. Substance molecule can fit together with the enzyme, stopping the reaction. Okay, so when the shape of the active site is no longer fitting this substrate, the reaction will stop. And this is known as denaturation for the active site, and this is permanent, so you can't go back to the shape of the active site as it used to be. In the shape of the enzyme, okay, once it happened, the enzyme will not work anymore, even if the temperature is reduced again to 37. Okay? Now going to pH. So the two things that affects the enzymes is the temperature and the pH. So we have two cases for pH. We have the acidic conditions and the alkaline condition, and we have the neutral. Okay? But some enzymes would work in acidic conditions and others can work in alkaline conditions, depending on its location in the body. Most enzymes work best at a particular level of acidity or alkalinity. You can call it acidity and you can call it alkalinity, or you can just simply say that it is pH. The protein digesting enzyme in your stomach, for example, will work at an acidity of pH 2, which is very acidic. At this pH, the enzyme amylase from your saliva cannot work. Amylase is found in your mouth, and in your stomach you can find, I guess, pepsin, I guess. Inside the cell, most enzymes will work best in neutral conditions. Okay? Inside the cell, it is in neutral condition. In your stomach, it's 2, which is for trypsin, and for amylase, it's another thing, okay? Because it's in your saliva and your mouth. The pH or temperature at which an enzyme works best is called the optimum pH or temperature, okay? Like the best condition is optimum. Conditions in the duodenum are slightly alkaline. So here they're alkaline, so they should be above 7. For instance, for pancreatic lipase, it is pH of 8. You need to memorize the pHs around your body. I guess I've mentioned it earlier in chapter 7. I'm gonna link it in the description box besides chapter 2. Although changes in pH affect the activity of an enzyme, these effects are usually reversible. Not like the temperature, you can just reduce the pH or increase it according to the enzyme, and then you can just get back to the active site as it used to be. Example, an enzyme that is disabled by a low pH will restart its normal active site when its optimal pH is met again. And you should know the shape of the graph. You need to know how to label it. So the x-axis is pH, the y-axis is the reaction rate, and here you should know that the very acid condition is pepsin. Most enzymes are around 7, and the pancreatic lipase is around 8. Okay? And that's all. Don't forget to subscribe for the channel, and see you in the upcoming video.